Hello everyone, my name is John Sunu. It's very nice to be here. I'm also very excited to be here. And this will be a guest online lecture for BME 514, year of 2024. I am a postdoc at Massachusetts General Hospital. And one of the excitements I have right now is that I will get to share you the methodology that I use every day, mostly every day, for all the clinical data analysis that I do here. The first part will be mostly the correlation analysis, and the second part is using a model to actually refine or clean the signal. And so we'll see how those methodology applies to clinical data analysis. I have a diagram here on the right. You can see this subject will be under pain because we are giving him the pain. In this case, it's a heat pain. You can see the temperature will be around some maybe body temperature. Then it would shoot up. It will be hot time to time in different frequencies and different durations and different magnitude, if you can tell. And under that circumstance, the subject will have a physical response, and one of them will be vasoconstriction response. So this is a laser Doppler flowmetry that is measuring the blood flow on the regional finger vasculature. And that is portrayed by this blue line. And you can see that when there's a heat pain, which you see in this red, which is actually flipped. So on the top, it's no pain, but when it actually in this direction, it is actually getting hot. You can see there is some kind of synchrony in terms of blue lines. But our question is, how do we objectively quantify how fast the response was, and how much, and how clear, and how large? So those will be the example for using a cross-correlation analysis. And on the second part, it will be model-based analysis, which I've used to actually clean the signal that I measured from FNIRS. FNIRS stands for Functional Near-Infrared Spectroscopy. And what's important is the clinical application. Why do we do this? Why do we quantify responses? So for example, in certain diseases, this is very important. How sensitive you are or how vulnerable you are to these pain responses will be very important. And also in the brain, it is important to measure how much brain activity actually was there, not just because you felt or you said ouch or I just feel miserable, because these are only very subjective. Okay, so we'll focus on two techniques. First one is cross-correlation technique with a real-life application. And of course, this is to objectively quantify any physiological response to some kind of stimulus. This will include moving window correlation, followed by a method called bootstrapping for statistical analysis to determine how significant your response was. Second part is dynamic systems model-based technique to filter out any signal contamination from other physiological confounders. And this was used to improve the functional near-infrared spectroscopy measurement, which indeed is known for those physiological confounders as a limitation. All right, so this is how I will demonstrate the techniques in two clinical applications. First one for the cross-correlation is to objectively quantify the pain perception in sickle cell disease, which I will go over what sickle cell disease is. And the second part with FNIRS is to actually objectively quantify the cognitive effort in sickle cell disease patients.
Now we'll talk about what sickle cell disease is about. It's a rare genetic blood disorder that is inherited from parents. And so for the healthy case, you see the red blood cells are very elastic, flexible, in a biconcave shape, and they can travel smoothly through the microcapillaries, exchanging oxygens and stuff. But in sickle cell disease, there is a mutation of the gene that's associated with the hemoglobin in the red blood cells that cause sickling when the red blood cells give out the oxygen. So as they become deoxygenated, you can see the sickling occurs here, and they can create occlusion. And the tissue around the occlusion can be deprived of oxygen, causing serious pain and can lead to a fatal organ damage. And even there's such no blockage, sickle cell disease patients are always anemic because they don't have many healthy red blood cells. So it is known that 1 out of 500 African Americans will have that, and 1 out of 10 African Americans will have a trait in terms of the genetics. Now, there are treatments, including chronic blood transfusion, medication, and bone marrow implant, but you can see that they do come with high risks. Altogether, when we look at the table for Centers for Disease Control and Prevention, you can see all these number of complications, including anemia, organ damage, and you can see that pain is listed. Pain is actually one of the most top reasons that people go to emergency room to take opioids, actually, to reduce the pain. Sleep disorder is here, and the stroke. So studies have shown that even with the treatment, a half of sickle cell disease patients would get silent stroke by age of 30. This is devastating because this is about cognition and development. This means, unfortunately, little is known about the cause and optimal therapy is needed.